Hello Biotechnicans. I am Dr. Farhan Zamir from Biotechnica, Bangalore. We have started a new series and this series is called as Ideas to Innovation. Now, this Ideas to Innovation series would contain a key concepts on how exactly our research on a particular topic could be actually perceived, and we will try to give you certain illustrations and ideas which can actually make you a very good researcher. So let's dive in. Welcome back. Today, we would start a beautiful series on ideas to innovation. So we will make sure that we will have particular illustrations of a great idea which could be perceived as a good research component, and this is how we are trying to mentor you on an individual basis, and let us see how best we can work together. Now, for today's topic, the topic for today is bacterial signaling and its mechanism, especially in biofilms. Now, to make you understand about this particular topic, I need to dwell more, and let us start with certain foundations. As we know, microorganisms are been coexisting with human beings from time immemorial. So, in these set of microorganisms, I have bacteria, fungus, parasites, viruses, etc. and etc. Now, what we will try to do is, we will take one niche of microorganisms, that is bacteria, and dwell deeper and understand what are its mechanisms for growth, survival and reproduction. Now, the first component is, when I talk about bacteria in its natural habitat can exist in two different forms. The first form is called as planktonic form and the second form is called as biofilm form. So what is planktonic form? Please remember, any bacteria will exist as a free-floating bacteria, and this free-floating bacteria is called as planktonic form. However, in nature, this free-floating form will not exist for a very longer time, and very soon there is a transformational change which will occur from a planktonic form to its biofilm form. Now, what are biofilms? Biofilms are aggregates of the same bacteria, but however, now this is being in a form of a mucilaginous layer, and within this mucilaginous layer, there is a heap of cells which will get accumulated. Now, please remember, these are also called as sessile forms. So, to have a quick recap, I have bacteria. Bacteria has two different forms. One is planktonic form and the second one is biofilm form. And remember, in most of the pathogenic cases, in most of the subjects who are suffering with one or the other kind of bacterial infection, it is 90% that the bacteria or the pathogenic agent or the bacterial pathogen will be in a form of its it will be in the form of its biofilm form and rather not in planktonic form. But also to remember, when I'm trying to mimic all these experiments in my laboratory, I will start with a planktonic form, and there are series of methodologies wherein I can transfer a planktonic bacterium into biofilm bacterium and then try to study the mechanisms. For this too, understood in a much better way, let us try to understand what exactly is the entire paraphernalia of the series of steps wherein a planktonic form could be converted into biofilm form. Let us try with the first step. The first step is the bacteria, which is in its planktonic form. Remember, it has a complete structure, that is the capsule structure. With that, it has flagella. Now, flagella is your planktonic. The planktonic form has the capsule, which is the main body, and the flagella, which is the locomotory organ. Now, what we need to understand is, as we humans, we look for certain permanent settlements. The same is the psychology of the bacteria. Now, the same planktonic form will now look for certain adherent surfaces. So once it finds an adhesive surface, this bacteria, which is in its planktonic form, will now try to come and sense the microenvironment. So it will decide whether I need to adhere onto this surface or not. And a point to be remembered is these bacteria are able to adhere for both biotic and abiotic surfaces. So the first component of the transformation is the planktonic bacteria will sense the adhesive surface. So this is the step number one. Now, once it thinks that this particular surface is favorable for its conditions, then the next component is the same bacteria will now try to adhere onto the surface. Once it adheres onto the surface, it produces certain extra polymeric substances, which we call it as EPS, or it is also referred as hexapolymeric substances. Now, when there is a formation of hexapolymeric substances, remember I told you I had the capsule and there was a flagella. At this point of time, the bacteria will decide to get from a motile form, it will get converted into a non-motile form. And then that is how it will adhere onto the surface and it will start replicating. Now, once it starts replicating, it is a factorial mechanism, and this mother bacteria, the first bacteria is called as mother bacteria, and this mother bacteria indicates the daughter bacteria or the other bacteria just to utilize the resources of the host and keep on replicating. And a very important key feature is the mother bacteria sends a signal saying that please do not produce any kind of toxin. Now, until the mother bacteria sends a particular signal, so there is no production of toxin from these particular daughter bacteria. Now, they are just replicating, and they are replicating within the host. Now, once this is the second step of growth. Now, once there is a sufficient amount of growth which occurs in that particular heap of cells, then these cells will undergo detachment, and upon detachment, they will go and get dispersed, and upon dispersal, that is how these new dispersed bacterial cells, which are still in their planktonic form or biofilm form, will now go and settle on an other surface, leading into secondary infection. Now, another important point what we need to remember is, as I told you, from a planktonic form, the bacteria has converted into biofilm form. Upon dispersal, the bacteria still can retain its ability to get converted from a biofilm form to a planktonic form, that is, from a non-motile version to a motile version. So, if we are trying to analyze the entire scenario of the story, so you now know that bacteria is much more intelligent than we actually understood it. Now, all this is general in case of bacteria, but let me try to give you a specific illustration which can actually help your research. Now, among so many bacteria, I will take a specific example of a bacterium which is called as Listeria, that is Listeria monocytogenes. Now, why is this bacteria so important? Now, whenever you're trying to focus on a particular research, you need to know the rationale of the study. And this rationale of the study will always define the rationale of why did you select this particular topic among the entire plethora of topics of life sciences. So here comes why am I selecting Listeria? Because Listeria is the model organism for facultative, intracellular pathogens. So, and it is of a major problematic in most of the food industry, and it has highest motility rate. Now, because of all these, factors, what we try to do is, we would decide that, okay, let me try to take up a bacterium which has a multifaceted effect, and also we will see that how relevant is this particular topic into two different domains. One is on a societal domain and the second one is on a scientific domain. So my kind of research should benefit both the domains of the society. Now, let us try to now dwell in more. So the genus Listeria has around 17 species, and among the 17 species, for your surprise, only 2 species are pathogenic and rest 15 species are non-pathogenic in nature. Now here comes a new question. The new question is, though I have 17 species, what made this 15 species non-pathogen? Only 2 species, that is Listeria monocytogenes and Listeria ivanovi, are the only 2 human and animal pathogen respectively. Now, what is this? This can be looked at either at a genomic level, at a proteomic level, at a transcriptomic level, or at an epigenetic level, okay, or at a metabolomic level. We can look into various purviews. So here it is able to cause the infection in humans, and please remember, Listeria causes infection in humans, especially in males. Okay, this condition wherein it is able to pass your brain, and this causes a condition which is called as meningitis. However, in specifically with females, Listeria is predominantly famous for creating multiple abortions. This is responsible for multiple abortions in females, and trust me, my dear friends, in India, we do not have a proper diagnostic kit which actually diagnoses Listeria, especially in reproductive system. Then this condition, which is, we call it as listeriosis, that is, though it is rare in India because most of the times we actually cook food well and then we consume it, but however, if any point of time, if you are infected with Listeria, then the mortality rate increases to up to 30%. Now, what are its growth and survival parameters? Now, please remember, as I told you, Listeria can actually survive in humans. So that means to say it will behave as mesophilic in nature. But however, this particular pathogen is also able to withstand minus temperatures, like it can withstand minus 20 degrees, and it can also reproduce at minus 8 or minus 4 degrees. So this is the colder part of it. So I'm trying to tell you that this microorganism can also survive as a saccharophile. On the other hand, this can also survive as a slightly thermophile, because this can also withstand a temperature of around 45 to 50 degrees. So this is a quite robust microorganism, and this is why I have picked up for my study. The next important component is, who are the people who are actually infected with listeriosis. Now, this could be widely categorized into a terminology which is called as YOPIs, Y-O-P-I. Y refers to young, O refers to old, P refers to pregnant ladies, and I refers to immunocompromised patients. So Listeria can have a wide approach on these YOP kind of population. Now, if this has to be understood well, then I need to always, remember to go into the sub-molecular level so that I understand the pathogen in a much better fashion. So let us try to understand the infection cycle. This is in courtesy of my German boss, Professor Jürgen Kreft, who actually painted this entire canvas of Listerial infection. Now, to start with, as I told you, Listeria is an intracellular pathogen, and it produces two interleukins, that is interleukin A and interleukin B, and this will help in actually invading the bacteria into the cell. So once it invades into the cell, it produces PLCA, that is phospholipase A with listeriolysin, and then the Act A of the bacterium gets activated, and that is how Listeria will travel from one compartment of the cell to another compartment of the cell. Now, finally, this produces various consortium of pathogenic cascades, and these pathogenic cascades are majorly controlled by a master transcriptional regulator, and this master transcriptional regulator is called as PRFA. So you can see the entire cascade of PRFA and how exactly there is an interdisciplinary conjugation or association between the pathogenic islands and that of the biofilm forming genes. Now, for me to understand this, I need to understand two major components. What happens in a biofilm form of a bacterium, what happens within the colony, and what happens into its outside the colony. So I am trying to understand the microenvironment which exists within the biofilm, and also the microenvironment which is surrounding the biofilm. So here I have two major components. Let us try to dwell upon the internal factors. The internal factors is, once there is a biofilm formation, there is an effective deployment of the cells which will take place. Then the frequency of formation is much more higher, and there is very, very slow penetration, and this can lead into an altered microenvironment. However, onto the external sources, onto the outer part of the biofilm, there is a huge amount of stress response, what we can look into. There is a huge amount of persistent formation which can result into antimicrobial effect, and also this can lead into various other nutrient utilization. So these are the important extrinsic and intrinsic factors which will actually govern the formation of biofilm. Now, when we were talking about stress response, what we did was, in terms of stress, how exactly a very important tripeptide, which is called as glutathione, which are normally the cytosolic scavengers, glutathione is nothing but it's gamma glutamyl cystinyl glycine. This is a small peptide. Remember, my dear friends. So, for all protein synthesis or peptide synthesis, there is the necessity of ribosome. But here is an example wherein you do not require ribosome for the synthesis of this particular glutathione. This simply happens with the conjugation of two important enzymes, that is GSHA, that is glutathione A, and GSHB. Now, this actually facilitates the entire mechanism. Now, what we are trying to do is, now I know that there is something which is called as biofilm forming genes. There is something which is called as virulent genes. Apart from that, I'm also looking upon how exactly the stress response, that is, the glutathione can have a profound role on biofilm formation gene and onto the pathogenic islands. So here comes an important pathway which is called as glutathione pathway. Apart from that, there are certain functional groups which play a important role with these particular, two important domains, and these are called as TDRM. Now, what are TDRM? That is thiol disulfide redox metabolism. Now, if you are observing the way the entire communication is going on, it is going deeper and deeper. This is because when we dig on deeper and deeper, that is how novelty actually blooms up. So hence, now from the entire stress response, I went on to glutathione, and now I'm looking into the entire glutathione family, which is called as TDRM, that is thiol disulfide redox metabolism. And we were able to come up with a set of genes which are interconnected, interrelated, so that we can study the gene regulation in a much better way. So these are the set of various kinds of genes which are responsible for biofilm formation, also for that of the pathogenesis. Now, to start with, you can procure Listeria monocytogenes either from ATCC or MTCC. ATCC is American type culture collection and MTCC is microbial type culture collection. Now, there are certain, because it's a pathogen, there are certain biosafety measures which has to be taken in your laboratory, and upon that, the culture can be procured from either of these particular agencies. So once you have them, please remember that would be in a form of a planktonic form. So once you develop them into a culture, that is the starter culture, now I want to study the various physicalities, okay, that is both physical parameter, chemical parameter and microbial parameter with this particular culture. So what I want to do is I want to study a simple factor such as what would be the effect of temperature on Listerial biofilm. So from a planktonic form, I will try to grow them in a beautiful methodology which is called as microtiter plate, and onto a microtiter plate, okay, I will try to develop my Listerial biofilms. But however, this development of biofilms, I am putting it on a range of temperature. So the range starts from 4 degree, 18 degree, 25 degree, 30 degree and 37 degree. And once I grow them, okay, you can look into all these micrographs. These micrographs actually, the A on the panel indicates 4 degree, B on the panel indicates 18 degree, C on the panel indicates 25 degree, D on the panel indicates 30 degree, and E on the panel indicates 37 degree. And F on the panel is how the biofilm looks like onto a membrane. So you can see this as a membrane, and onto the membrane, how exactly the layer of biofilm is actually flipped. Okay, and this is the thickness of the biofilm. But when I look into it, you can also look into certain structures which are thread-like, and these thread-like structures are nothing but your EPS, that is hexapolymeric substances. Now, as we have understood the pure culture formation of biofilm, we can also extrapolate the story for the mixed culture. So if you are interested with, understanding the consortium of bacteria, that is from Listeria, and how exactly Listeria can grow with Staphylococcus, or Listeria can grow with Pseudomonas, Listeria can grow with Helicobacter pylori. So all these kind of, mixed culture biofilms could be also studied. Remember, my dear friends, the methodology still remains the same, but the only thing is the permutation and combination of the different pathogens or different bacteria has to be designed by the researcher. Now, once we looked into the entire genes, once we have sorted out the entire genes, now what we have done is using bioinformatics, we now know how exactly is the structural component of the TDRM looks like, whether it is alpha, alpha prime, or alpha, beta, or beta, beta prime, all these kind of orientation of proteins on a domain which is called as CXXC is noted. CC refers to disulfide cysteine and XX could be any of the amino acid, and for a canonical thyrodoxin, this XX should be GP, that is CGPC. Now, when I look into these kind of specialized genes, what we have done is in Listeria we can also go upon the mutational studies. Now, when we do mutation, either it could be an ins mutant, that is insertional mutant, it could be a delta mutant, which could be a knockout mutant, or there could be a silencing cascade, which could be still employed. And once we have done all these kind of gene expression studies, you can see that there is a various kind of pathogenic islands which actually exist between the mutants what we have studied, and this profiling becomes very, very important. Now, since we have studied on a small set of mutants, now if I want to study on a global pattern, the best method is to go for global transcriptional analysis, okay, of thiol genes of using DNA microarrays. Now, once we have done this, we actually screen for around 2000 or 2500 genes, and among them what we have observed is there are more than 300 genes which are actually upregulated by 2 folds, and there are more than 150 genes which are actually downregulated by 2 folds. Now, taking this as a criteria, as a threshold baseline, now what we try to do is we will try to sort out that which are the actual genes which are involved and which can play a predominant role in the virulent cascade island and in the biofilm forming island. Now, once we do that, we are able to come up with the mode of action in explaining the mechanism of how exactly this biofilm formers, especially the Listerial biofilm formers, play an important role and how are they different with that of the planktonic form of the same bacterium. Now, if you are doing with the Listerial component as a pure culture, this is fine. Or if you want to again extrapolate the data, you can also study with the mixed culture biofilms. Now, when you put up a lot of hard work, you knowingly or unknowingly, you start up accumulating data of a particular organism, and this actually came as a surprise for me when we were working on Listeria, and somebody actually wrote about the researchers who are working on Listeria and on Listeriosis, and for our surprise, now we know that it was only 5 groups in India who are actually focused on this pathogenesis, which is called as Listeriosis, and our group was one among them at University of Mysore. Now, the learnings from Listerial research could be extrapolated to another important bacterium which is called as Helicobacter pylori, which is mainly responsible for causing peptic ulcers in humans. So, what I'm trying to make you understand is all this is relatable, and it doesn't matter that what organism you take, but however, there should be a small amount of novelty which has to be established during your study. Now, what we did was, as I was trying to tell you, there's a beautiful communication between the mother bacterium and the daughter bacterium, and then one daughter bacteria with another daughter bacteria. And this is through a beautiful networking, and this networking is called as hexapolymeric substances or hexapolysaccharides. Now, what would be the strategy for anti-biofilm mechanism? Now, until now, we spoke about biofilms, but what are anti-biofilm strategies? Now, the anti-biofilm strategies could be of 2 major types. One, I can actually have a bacteriostatic mechanism, or I can have a bactericidal mechanism. Now, if I want to have a bactericidal mechanism, what I do is I kill the bacterium. Okay, somehow I make sure that I add either by chemical method or by phytochemical method, I'll be taking these molecules and I will be actually disrupting the entire bacterial community. This is called as bactericidal phenomenon. But however, if I'm planning for a bacteriostatic activity, then the other strategy is, let me try to take up a ligand molecule and then inhibit the formation of the EPS. So that once I inhibit the formation of EPS, one bacterium cannot communicate with another bacterium, and that is how I break the communication between one bacteria and other bacteria, and this kind of a communication is called as quorum sensing. So I am actually designing certain molecules which are anti-quorumone molecules, and if these molecules are from a that is from a plant source, then I can call them as phyto-antichromones. Now, if you are taking these molecules and then developing a nanomolecule out of it, which can show an anti-choromone molecule-like phenomena, then I can call it as nano-phyto-antichromones. So all these are new terminologies which are emerging in terms of bacteriology. Now, all this research was being done with the guidance of great people. My guide, Dr. Shubha Gopal, who is a professor at University of Mysore. Then Professor Krone, who taught me electron microscopy, all SEM and TEM, and he at University of Würzburg, taught me one important thing is, if you have patience, you can get whatever you want under the microscope. The other group was from University of Tel Aviv, Professor Yahya's group. And then I have my three friends, were Dr. Vani, who was my senior when I was pursuing my PhD, and then Dr. Megashri and Dr. Sangeeta, who were my contemporaries, who actually helped me out. And obviously, the man at the center is Professor Jürgen Kreft, who was instrumental in teaching the entire mechanism of bacterial pathogenesis and virulence. And with all these great people, I was being mentored. Now, the intention of telling you all these things is, my mentorship was with the hands of the giants. Now, please remember, if you want to get trained, and this is only possible by very good mentorship, and this kind of good mentorship, has been mainly facilitated by Biotechnica. So just make sure that for any kind of assistance in science and research, please get assistance from Biotechnica, and we'll be very, very happy to translate your ideas into innovation. That is what I wanted to tell you for today. Thank you very much. Stay connected. Happy science doing.